I will start now the official. Like, this is the second edition of this poster-cat meeting here at the Institute of Catalysis. As you can recall, we are just gathering all together here, or as much people as possible, to show all your results that you have been presenting through the whole year in different conferences. So then we can learn from each other and we can share our knowledge and see if maybe there are future opportunities for collaboration between each other, and everything goes smoother and we make more friends here in the Institute. And we don't stay everyone in their own room just focusing on their things, which is a little bit lonely and sad. So that's the point. So we have a nice time together. Later I will write by hand, written in blood, with all your certificates, so you can happily say, hey, I got this fancy line in my CV, and everything will be okay. I would like to point out that this is my first act talking as a director of this institute. So thank you very much for all your support, and just tell all the most young people that is here that 20 years ago I was sitting there. I would have been sitting there if there was an act like this in that time, and there wasn't. We just make different sort of granted fellowships, coffees, on these things. So this is more official, and I think it's an upgrade. So I'm very happy about it. So I think that I have already put here all the things I want to say. So we can start like 3 minutes early if you want. We can start because probably the first speaker want to come here is Yahan Wu.
We may just very briefly comment about how this will proceed. So we have a session from 10 till 11:30, and each speaker has 6 minutes from the seat to the seat back. So it will be very fast, and there is no time for questions. The questions are reserved for the coffee break and the lunch afterwards. And so Marcos and me will chair this first session. And now the floor is yours. Yahan Wu will talk about the directed evolution of peroxygenases and for hydroxylation of tyrosol.
Sorry. Good morning, everyone. I'm Yahan Wu. My topic is directed evolution of peroxygenases for hydroxylation of tyrosol. And I will start with the hydroxytyrosol. It can be found on olive oil and olive leaves. It has antioxidant properties. It is widely used in food and pharmaceutical industries. It has a great pharmacological potential like antifungal, antibacterial, antiviral, and several methods have been used for obtaining this substrate. However, the extraction method has a low extraction yield and extensive amount of the organic solvent. The chemical method has a complex procedure and hazardous reagent and harsh reaction conditions. In contrast, the biological method it's more economical, effective, and environmentally friendly. But the biological method described in the literature has low yield, and the production efficiency of the hydroxytyrosol is not suitable for the industrial production. So it is necessary to find out the high effective tyrosol hydroxylase. And we use tyrosol as the reaction substrate because the tyrosol has the similar structure with the hydroxytyrosol, and it can be obtained from the cooking waste. Also, we use the unspecific peroxygenases at the biocatalyst. It can hydroxylate the aromatic compound, so it could be a considerable tool for the tyrosol hydroxylation. So the objective of this study is use UPO as the biocatalyst for toluene radioselective hydroxylation of tyrosol by directed evolution. Now we talk about the directed evolution. Directed evolution is the protein engineering method, and it's mimicking the process of Darwinian evolution at the test tube. And this is the workflow of the directed evolution. It starts with the parent genes, and we create the mutant library by aeropron or DNA recombination, and then we transfer all these genes to host to produce protein, and we pick up the colonies in 96 well plate to express the enzyme. Then we use those enzymes to screen and to find out the best mutant. The best mutant can be used as the parent genes for the next cycle of the evolution. To facilitate the library screening, we designed a high-throughput screening assay. It's based on a formal publication. We adjust that protocol to our own project. And this is the result of the first directed evolution round. And we create the mutant library by aeropron PCR, and we screened 1,000 colonies. For tyrosol hydroxylation, the best variant has a bifold activity of the parental tab. And also we noticed that the activity of tyrosol and the activity of ABTS, there's no correlation between those. It means we evolve enzyme for tyrosol hydroxylation. It's basically removing the activity of against the ABTS. So in conclusion, we established a high-throughput screening protocol for exploration of the tyrosol hydroxylation activity in peroxygenases. We prepared a library by aeropron PCR of the 1,000 colonies. We obtained a set of improved variant for tyrosol hydroxylation. This directed evolution workflow will be repeated several times, aimed to obtain improved variant for the production of hydroxytyrosol. Then we will investigate the expression, purification, and characterization for the best final variant achieved. And that's all. Thank you very much.
Thank you. So it was very fast. Thank you. The next speaker will be Pedro Manuel Abellanas Pérez, and he will talk about co-immobilization of enzymes, different stabilities, using heterofunctionality of reduced animated support modified with glutaraldehyde.
Thank you. Okay, so hello, thank you for the introduction. So I'm Pedro, and I'm going to be talking about co-immobilization of enzymes with different stabilities using heterofunctionality of reduced aminated supports modified with glutaraldehyde. To start, co-immobilization is basically when you immobilize two or more enzymes in the same support. An issue that can happen when we do co-immobilization if the two enzymes have different stabilities. When this happens, one of the enzymes can get inactivated before the other one, and this first one remains fully active. When this happens and they are immobilized by the same method, we only have two options. If they are immobilized by a reversible method, we can detach the enzyme from the support, and with some fresh enzymes, we can reuse the support. And if they are irreversibly immobilized to the support, our only option is to discard the whole biocatalyst. In both cases, we're discarding fully active enzymes, which is not ideal. So that's the main focus of this work. So we used two enzymes as models. The first one was Eversa Transform 2.0, which is a lipase, and then a beta-galactosidase from Aspergillus oryzae. As you can see in this graph, they have different stabilities when incubated at 55 degrees and pH 8. So we knew that we wanted to use aminated supports and modify it with glutaraldehyde, but we had two ways to go about it. The first one was to immobilize the first enzyme via ion exchange to the support, and then modify it with 1% glutaraldehyde, which would allow for monomers of glutaraldehyde to modify the enzyme and the support. Then we would reduce the glutaraldehyde to be able to immobilize the second enzyme via ion exchange. The other option would be to pre-activate the support with 10% glutaraldehyde, which would allow for dimers of glutaraldehyde to modify the support, and then we could covalently immobilize the first enzyme to the support. And again, after reduction of the glutaraldehyde to take away its chemical reactivity, we could immobilize the second enzyme. Whether to choose one or the other method would come down to whether we were able to release the second enzyme. So we incubated the biocatalyst in different concentrations of sodium chloride, and as you can see, and then we, after thoroughly washing the biocatalyst after incubation, we did SDS page analysis, and as you can see in the first analysis, we were able to detach the enzyme from the support, the 1% glutaraldehyde support with 0.3 and 0.6 molar of sodium chloride. But when using the 10% glutaraldehyde support, we weren't able to detach the enzyme from the support using any concentrations. So we went ahead and, and chose the first method for that reason. And we tried to reuse cycles. And in this graph, you can see the solid squares are the residual activity of Eversta, and the triangles, the beta-galactosidase. The red arrows indicate the point where the beta-galactosidase got inactivated, and we desorbed the enzyme and added fresh enzyme to the biocatalyst. And as you can see, Eversta didn't lose any of its activity throughout the process. And so here you can see the strategy that we propose for when this issue or this problem comes. And that's basically it. Thank you so much for your attention.
Thank you very much, Pedro.
So our next speaker will be Gabriela
Cantaragio, yes.
From the group of Dirigida de Enzimas, de Miguel Alcaide, and he will speak about directed evolution of fungal peroxygenases by dystrophic display.
So, hello everybody. My name is Silvia. I'm a PhD student at Miguel Alcalde's lab, and today I'm going to speak about directed evolution of fungal peroxygenases by surface display. So, first of all, I'm going to introduce these enzymes, although my lab mates have already talked about this, but I need to mention it. Well, these enzymes, they are able to catalyze the oxyfunctionalization of non-activated CH bond, which is a reaction with pharmaceutical and industrial interest. They use the hydrogen peroxide as the main oxygen donor and the final electron acceptor. And they are secreted into the extracellular media, so we can work with supernatants. And among their weaknesses, we can mention their low heterologous expression, the undesired peroxidase activity, and the oxidative inactivation provoked by their co-substrate. So here we have the first UPO that was expressed in the lab. It comes from the fungus Agrothidae aerita, and this enzyme was subjected to directed evolution, and as a result, pattern mutant was obtained with a better functional expression and improved kinetic parameters. So, well, this is Darwin riding a turtle, in case somebody is wondering who's this guy. So how was that process done? Well, my lab mates have already talked about it, but again, I'm going to repeat in order to compare this workflow with my new one. We start from the parental genes, we create the mutant library, we clone and transform the yeast, then we pick the colonies one by one into these 96 well plates, and finally, using robotic platforms, we perform the screening assay, which in this case is based on a colorimetric test. So with this workflow, we can analyze between 1,000 to 10,000 variants per round, with between 1 to 3 mutations each variant. So this is quite a lot, but we are leaving behind so many variants untested. So in my PhD project, what we want is to introduce new generation technologies in order to reduce the screening effort and increase the throughput. So here, instead of working with enzyme in supernatant, with that process of picking the colonies one by one in that 96 well plate, here we are going to express the enzyme attached to the surface of the yeast, and we will use anchoring systems like, for example, the fusion protein. So if the enzyme is well expressed, as a result of the peroxygenase activity on this substrate, the biotin phenol, it will appear on the surface of the cell, these biotin-related residues. And if we add to the reaction this tetrabidim conjugated with a fluorochrome, then we will be able to detect the enzymatic activity using techniques that measure fluorescence, such as flow cytometry and FACS. So here we will have a sense suspension in one single tube that will contain the entire mutagenic library. So as you can see in this figure, the cells, they pass one by one in this continuous and linear flow in front of a laser beam. So if they emit fluorescence, then the cytometer, it will be able to identify them and count them. And if we have the FACS technology, we can sort or separate the cells, and at the end of the process, we can collect them, so we can keep our winner mutants. So this is the throughput of this workflow. We can analyze between 10 to 6 and 12 variants per run, with between 5 to 8 mutations each one. So it's quite an improvement. And having in mind that this is an automated process and super fast, it will be really interesting to introduce these technologies to our experiments, and we are looking forward to see how it goes. So thank you to my group, and thank you for your attention.
Thank you very much, Silvia. Next speaker is Fadia Cervantes Domínguez from Systems Biotechnology Group. Fadia will explain us about biologics in synergy to degrade target macropollutants. Thank you. So start whenever you want.
Good morning, I'm Dr. Fadia Cervantes, so I'm going to tell you some results about NYMPHE project. Well, about 3,500 chemicals are proven that has ecotoxicological influence in this polluted nature that we have now. In this case, we have, for example, biocides, plastics, or pharmaceuticals. And one of the examples that we have of this pollution is how the pharmaceuticals are entering in the water cycle. So here we can see that first the production in the pharmaceutical industry. Here they have a lot of normativity to try to not have a improper disposal, but they have some improper disposals, and we can find also the compounds, untouched compounds in the wastewater treatment. Well, in the other side, we have like the distributions to the pharmacies, and then the disposal from people that can be proper disposal or improper disposal, like flushing the pharmaceutical compounds in the bathroom, for example, and it arrives also to the wastewater. So then we have the treatment of these wastewaters, and it's very difficult in the different steps of filtration to take out the drugs and the metabolites of drugs. So there's a lot of new health problems that the UNESCO started to bring in different studies, and in 2017 one of these studies showed that only 9 of 118 compounds are removed with efficiency in 95%, and half of the compounds were removed only partially in 50%. So because of this, there's a lot of interesting investigations that are talking about how to how it is entering in the drinking water and causing a lot of health problems in people, and how to remove these compounds. So in INFE project, we have like the objective to create and develop bioremediation strategies using enzymes and other strategies to try to take out these compounds at least in 90% of efficiency. So in the part of the project I am working is in the degradation of different pharmaceutical compounds. We started using the search of references to see the degradation pathways. Then we have metagenomes, and we are searching some enzymes to degrade these compounds and to create the enzymes in the laboratory and then test with these drugs. So the first drug I started to work with is the enalapril, that is an angiotensin converting enzyme inhibitor. And the first reaction I am studying is the hydrolysis of the ester to create the enalaprilat. So I tested 23 different esterases, and I find more or less 5 enzymes that could be possible to hydrolyze this ester. And then I try again these 5 enzymes with less amount of enalapril, and I found that only the yeast-14 has the activity to change the enalapril to enalaprilat. So I started to see the best reaction conditions, and I saw that the best activity was at pH 9 and 45 and 50 degrees. So I decided to use 45 to try to have more stability of the enzyme. And then I tried 3 different mutants for the yeast-14, and I tried the same reaction conditions that I used for yeast-14, and I have good results with the Y334S. Then I put less amount of enzyme to compare these 2 enzymes, and I found that it's better, the activity is better with the Y334S. So I will continue my experiments with this. That's all. Thank you.
Thank you, Fadia. Our next speaker will be Nicolás Coca López from Spectroscopy and Industrial Catalysis Group, and he will talk about data science for Raman spectroscopy.
Okay. Thank you for the introduction, and good morning, everyone. So I would like to start the presentation by discussing what is... what is it? Joder. Vale. Vale, gracias. No sé cómo enganchar esto. ¿Se me escucha? Sí, vale, guay. Gracias. Vale, bueno, pues I would like to start Eso, ya tengo un cacao que esto no es... I would like to start the presentation by discussing what I mean by data science. So normally it is said to be based in 4 different pillars. The first one could be the field knowledge. So this could mean either, I don't know, business, economics, finance, or in our case, Raman spectroscopy. Then it could be a combination of mathematics and computer science, meaning by this algorithm development, knowledge on databases, cloud, and so on. And last, storytelling. So the idea could be that you get your data, play with it, and then be able to tell a story based on that. That eventually would help either your research, your company, whatever. So an example of Raman spectroscopy data pipeline or workflow would be the following. We collect the data, storage it, format it, whatever. Then it is processed following several of these different pre-processing steps. And then we model the data. That would mean either statistical modeling or some modeling based on machine learning, like regression, classification, or clustering algorithms. So in principle, machine learning can be found in every of each steps of this pipeline. So in this context, I have been working the last 2 years in this European funded project called Charisma, where we try to develop different protocols for calibration and standardization of Raman spectroscopy. And in the following slides, I will try to show you what we have been doing regarding that. First of all, we develop a Python library called Ramanchada 2, which is open source and that can help with the different preprocessing steps, as would be spike removal, baseline subtraction, smoothing, peak fitting, and of course calibration. The first of these steps is normally spike removal. For this, we have developed an in-house algorithm in order to get rid of these spikes. Spikes are these thin peaks in there, which happen when a cosmic ray hits the CCD detector, and therefore generating these small peaks that then affect the processing afterwards. So not happy enough with that, we try to develop a full family of algorithms, implement them in this open source library, and also implement some traditional ones. The idea behind this was that normally people, when they publish their algorithms, they don't publish the code, and normally they also don't compare their algorithms to the previous ones. So we thought that we could also do that and provide a couple of tools to do this. One of them based on big data, so synthetic generated spectra. So it's so we are able to compare the algorithms based to this, and for example, give a F1 score metric, whatever. Then we were looking at different materials for calibration, and we choose some others: neon, silicon, calcite, and polystyrene. And the first thing we found in our interlaboratory study is that the peak shape affects a lot the peak fitting. So then we were able to recommend different and specific peak shapes for the different materials to do that, and also come up with a minimum signal-to-noise ratio, which would be necessary to this to be working effectively. Besides, we are working also in a couple of publications based on statistics on the results that we found on the different materials. And finally, I would like to give you a couple of examples on what is data modeling of Raman spectra. The first one would be a classical one, in which we use classical least squares to model or to monitor the intercellular light conversion from Fadia side to yeast mondaine, and correlate that with XRD data. On the other hand, we could also use machine learning modeling, in this case to classify leukemia subtypes with the help of Raman spectroscopy. And in this graph, for example, you can see how the different algorithms perform in front of an F1 metric for the different processing pipelines. So with this, we can learn that it's not only the algorithm that matters, but also the preprocessing of the spectrum. And with this, I would like to thank my colleagues and collaborators, and of course you for your attention.
Thank you very much, Nicolás. So we are reaching the half of the session. So first of all, thank you very much for keeping a schedule. You can relax the next 8 speakers. And I want to also tell you that, as you know, there is a parallel event going on in-house that is regarding this photography contest we have. So I have been told to tell you that you can go just during the coffee break or just before to vote. Okay, it's in the underground. So that's it. Next speaker, please, is Iris Cuesta Mayorga, from the group of conversion of carbon dioxide and circular chemistry, talking about perovskites. I cannot do 3 things at the same time.
I can present the figures.
I have to adapt to this. Okay, so.
Vale, ¿se me oye?
All yours.
Vale, sí. Vale, buenos días. Voy a presentar parte del trabajo que llevamos haciendo un año y que se engloba dentro de un proyecto que trata de fabricar celdas solares basadas en perovskitas. Estas celdas solares tienen que ser sostenibles, estables y eficientes. Para que sean sostenibles hemos realizado mecanosíntesis, es decir, una síntesis que no necesita disolventes, por lo tanto eliminamos todo lo contaminante de esa parte, y también que sean libres de plomo o disminuir el contenido de plomo de estas perovskitas. Como estas celdas solares van a estar en contacto con el oxígeno y se van a calentar, era necesario también hacer un estudio sobre la estabilidad frente al oxígeno y frente a la temperatura. Y por último, en cuanto a la eficiencia, es necesario hacer medidas electroquímicas, para lo que hemos tenido que depositar estos materiales en un sustrato. En cuanto a la síntesis, hemos logrado sintetizar estas 8 perovskitas mediante mecanosíntesis en un molino de bolas de carburo de tungsteno en las condiciones que aparecen en la tabla. Posteriormente, la primera caracterización que hemos hecho son difracción de rayos X para ver la estructura que tenía. Y a partir de ellos hemos determinado el tamaño del cristalito. Además, también hemos hecho una caracterización textural para conocer el área superficial, el volumen de poro y el tamaño de poro. Y también una primera caracterización ultravioleta visible para conocer el bandgap y para conocer, por lo tanto, cómo de fácil va a ser ese salto electrónico que va a generar la corriente. En cuanto a la estabilidad, en primer lugar aquí muestro el estudio de la estabilidad frente a la temperatura, para lo que hemos hecho análisis termogravimétricos que se observan en las gráficas de la derecha, en atmósfera de aire la de arriba y en atmósfera de nitrógeno a la derecha. Como se puede observar, las dos líneas, la verde y la rosa, que corresponden respectivamente a la perovskita de cloro y de yodo, pierden más de un 1% de su masa a unas temperaturas inferiores de 150 grados. Por lo tanto, las vamos a descartar por no ser suficientemente estables. En cuanto a las perovskitas cuyo catión B es exclusivamente plomo, las vamos a descartar pese a su gran estabilidad, precisamente por eso, porque tiene plomo y uno de los objetivos del proyecto es disminuir este metal del compuesto. Y por último, en cuanto al resto, se observa cómo en aire se forma una única fase, o sea, tras la síntesis en nitrógeno y expuestas al aire se forma una única fase al comienzo, pero sufre una transformación normalmente a temperaturas iguales o superiores a 150 grados, salvo la perovskita de cesio y bromo con una sustitución parcial de plomo por estaño, pero esa transformación no tiene por qué ser mala en un principio. Y en cuanto a la estabilidad frente al oxígeno, observamos que justo las perovskitas que tienen exclusivamente plomo y las que he mencionado antes, la de cloro y la de yodo, son tremendamente estables. No solamente se pueden sintetizar tanto en aire como en nitrógeno, sino que aguantan perfectamente tras un mes de reacción con la misma fase inicial. En cuanto a la perovskita de metilamonio bromo con una sustitución parcial de plomo por estaño, se puede sintetizar también en ambas atmósferas, pero evolucionan a lo largo del tiempo. Y en cuanto al resto, si queremos obtener solamente una fase inicialmente, solamente las podríamos sintetizar en atmósfera de nitrógeno. Por tanto, a modo de resumen, aquellas perovskitas que pierden menos de un 1% de su masa a una temperatura inferior de 150 grados y hemos logrado disminuir o eliminar el plomo son las siguientes. Si las sintetizamos en ambas atmósferas, hemos conseguido tomar como válidas la perovskita de metilamonio bromo con una sustitución parcial de plomo por estaño y sintetizándolas solamente con nitrógeno, esa misma pero con una sustitución total de plomo por estaño, y las equivalentes con cesio en lugar de metilamonio, todas ellas durante cortos periodos de tiempo. En cuanto a la deposición de estas perovskitas sobre un sustrato de FTO con una concentración de 7 miligramos por mililitro, hemos probado 3 métodos: la de electrodeposición, que aparece en la zona azul abajo a la izquierda, el drop casting y el deep coating. Hemos comprobado la homogeneidad de estos recubrimientos mediante microscopía electrónica de barrido y podemos observar que la electrodeposición no es válida en nuestro caso, ya que prácticamente no se deposita nada o directamente nada. El drop casting es un método muy rápido, se deposita muy bien, lo que pasa es que los recubrimientos no son todo lo homogéneos que nos gustaría. Y por tanto el deep coating es verdad que se deposita menos, pero sin embargo estos recubrimientos son muy homogéneos y la técnica bastante reproducible. Por último, mostrar que también realizamos medidas electroquímicas. Aquí se muestra datos de cronoamperometría en la que registramos la intensidad de corriente generada a lo largo del tiempo. Entonces, esta técnica nos da una información sobre cómo de rápido decae esa corriente y cómo de alta es esa corriente a lo largo del tiempo una vez que se estabiliza. Y también voltamperometrías, en la que nos da información acerca de los procesos que tienen lugar al variar el potencial. Cada banda que aparece sería un proceso de oxidación o reducción. Estas últimas partes de electroquímica todavía estamos en ello. Y por último, agradecer tanto al proyecto que financia como a la unidad de apoyo que ha hecho todos los análisis.
Gracias, Iris. Thank you.
Next speaker is Estefania Fernandez. Thank you very much, Christian. This microphone is killing me.
Can you hear
next
me?
Speaker is
Yes. Okay.
Estefania Fernandez, modeling for theoretical catalysis group, right?
Yes.
So here you go.
Yes, I'm Estefania.
Whenever you want.
Oh, sorry. I'm Estefania Fernandez Villanueva. I'm a postdoctoral researcher from the group of Veronica Ganduglia and Pablo Lustenberg. And for this work, we also work with Rubén Pérez from the Universidad Autónoma de Madrid. And I will tell you a bit about methane to methanol catalyzed by palladium gold nanoparticles supported on Syria. Okay. So you know the reaction, methane to methanol. It's interesting because it removes methane, which is the second most abundant greenhouse gas, and you convert it into methanol, which is useful directly as a fuel, but also as an important precursor in industry. But as usually happens, this reaction has competitive reactions. And so the idea is that methane is a very stable molecule, and once you find a catalyst that is able to break the first carbon-hydrogen bond, then the resulting the resulting CH3 species is not that stable and tends to overoxidize to CO or CO2. Okay, so you need to find a catalyst that is able to stop here at CH3. Okay. So we also collaborate with the experimental group of José Rodríguez, and they found this is an XPS of carbon 1s, and these two peaks are precisely related to what I was telling you about. So if you see this peak, it means that you have overoxidized methane and you lose selectivity, and if you don't have this peak, it means that you are not able to activate the molecule, so you are not doing anything. So when you have palladium nanoparticles on Syria, then you see both peaks, so it means you do not have selectivity. It is too active, but it doesn't have the selectivity that you want to methanol. If you use gold nanoparticles on Syria, you have no activity whatsoever. But if you mix the two, you can find the specific ratios for which it is selective to methanol. And the amount of palladium is relatively low compared to gold. So the idea is that gold is somehow limiting the dehydrogenation power of palladium. So in order to study this, we wanted to study the reaction mechanism on a bimetallic nanoparticle. But to do that, you have to first come up with a model, and we used a scheme based on something that was already reported. So we used two computational techniques, the conventional DFT optimizations with VASP that you probably know, but also we used the Goffey algorithm. It is an evolutionary algorithm, which means that it does not only generate a lot of random structures, but it also directs the search using first principles, because it starts from a population and does DFT single point calculations, and then builds a machine learning model that maps the energy landscape, so that when you build subsequent random structures, you can use this energy landscape to relax without using DFT calculations, so this is very fast. So the idea is that using Goffey, you can explore many more structures than you would do using only DFT. But it has a problem, and it is that it doesn't treat electrons. So when you put nanoparticles on Syria, metal nanoparticles on Syria, the metal nanoparticle gets oxidized and electrons go to the support. And so cerium 4 plus atoms from the Syria become cerium 3 plus. And you do not have that here. You can put them, but you have to select them manually beforehand. So you need to do subsequent runs of different Goffey runs and VASP optimizations to obtain your final model. But eventually, we found that most of the structures, in agreement with the experimental as well, in most of the structures, the palladium atoms get covered by gold, and you have a very few atoms accessible. So in this one, this is the most stable structure. You only have one, and it's the one that the thing that you obtain in most of structures. But we manually selected another more unstable structure to compare with it, that has two palladium atoms accessible, forming a dimer. And with these two models, we studied the reaction mechanism, and I don't have time in 6 minutes to tell you everything about it, but one key result is that when you have this palladium dimer, you are able to stabilize this CH2 species, and therefore you are favoring the competing reaction, which is what you do not want. In contrast, when you have only one palladium atom, this becomes greatly unstabilized both kinetically and thermodynamically, and that's how you increase your selectivity to methanol. So I will be by my poster if you want to know more about this. And with that, I thank all my colleagues, the funding, and you all for your attention.
Thank you, Stefania. Our next speaker will be Dr. Los Angeles Sánchez Pérez from Energy and Sustainable Chemistry. And he will talk about formic acid-mediated catalytic transfer hydrogenation of malic acid to succinic acid, and the role of the activation treatment of the palladium carbon catalyst.
Good morning, everyone. Thank you for the presentation. As they say, I'm Victoria, and I'm going to show you my work about the catalytic transfer hydrogenation of malic acid to succinic acid. So first, all of us know that some of us are working to reduce the use of petroleum and its derivatives, and for that reason, biomass have attracted growing interest because it's a sustainable and clean energy source. Because from biomass we can produce biofuels and different chemical products, such as 1,5-pentanediol, sulfuric alcohol, malic acid, and others. But in this presentation, I will focus on succinic acid. This product has many applications in the chemical industry because with that product we can produce 1,4-butanediol, cosmetic, and some polymers, such as polybutynyl succinate. And with that polymer we can produce a lot of products for packaging, automotive, and others. But currently, succinic acid production is mainly based on the hydrogenation of malic acid derived from petroleum. But nowadays, several routes are being developed to turn succinic acid into a renewable monomer. So the petrochemical route can become renewable if both the malic acid and the source of hydrogen necessary for the catalytic transfer hydrogenation are derived from biomass. And in our case, we choose formic acid as source of hydrogen because we can obtain that product from biomass, and we can also obtain malic acid from biomass via oxidation of gas phase oxidation of sulfur. So in this work, we report the catalytic transfer hydrogenation of malic acid to succinic acid using formic acid as source of hydrogen and palladium carbon as catalyst. And I'm focused in the effect that using different activation protocols of the palladium carbon system may have of the palladium species present in the catalyst and in the catalytic activity. So first, we prepare the palladium carbon catalyst by wet impregnation, and then we carry out several activation protocols such as calcine the catalyst in nitrogen, calcine the catalyst in nitrogen and then reduce with hydrogen at 2 different temperatures, or calcine the catalyst in nitrogen, then reduce in hydrogen and finally passivate, or calcine the catalyst in air or reduce with hydrazine. So in this graphic, you can see the succinic acid formation rate, and we can observe that we obtain the best results with the catalyst reduced with hydrazine and with the catalyst reduced with hydrogen at 343 Kelvin. So these results suggest that the species forming in the reaction process play a role in the catalytic activity. For that reason, we decided to carry out different characterization techniques, and I'm going to explain one of them. We characterize the catalyst with X-ray differential analysis, and we characterize the fresh catalyst and the used catalyst, the catalyst before the reaction and post-reaction. And when we characterize the fresh catalyst, we observe significant difference in the relative proportion of the palladium phases that can be present in that catalyst, palladium carbide, metallic palladium, and palladium oxide. And when we characterize the used catalyst, we can observe several changes in these phases. And the most significant change is observed in the catalyst calcine in air, because we can observe that the metallic palladium and palladium carbide phases rise at the expense of palladium oxide. So with this characterization, we can conclude that the reaction medium modifies our catalyst. And then we also decide to reuse the catalyst. We reuse 4 different catalysts, and in all cases we can observe that the reaction medium doesn't deactivate the catalyst, and in this case we can observe a slight change in the catalytic activity. So here we can observe that these catalysts are very stable. So to sum up, we can conclude that the different activation treatment affect the palladium phases that can be present in the catalyst and also the catalytic activity. The reaction modifies these palladium phases, and these catalysts are have a good stability. But we need to make more characterization to understand the role of the metallic palladium and palladium carbide phases in the catalytic activity, and for that we are carry out several characterization techniques such as XPS characterization. So thank you all of us.
Thank you very much, Victoria. Next speaker is Alicia Gomez from Spectroscopía and Catalysis Industrial Group. And we're going to see some biocidal activity.
¿Se escucha? Sí, vale. Thank you for the introduction. So, airborne infection diseases are mainly transmitted by aerosols contaminated with pathogens that remain infections for hours. This infection probability this infection probability is much higher in closed spaces where we spend more than 19% of our time. Nowadays, many technologies of purification and or inactivation pathogens present problem of low efficiency or high energy consumption. For these reasons, in this work, polymeric catalytic filters have been coated with an biocidal active phase, which will not release reactive species and hardly need for replacement because they operate inactivated rather than trapping. The objectives of this work is the preparation of polymeric catalytic filters by immersion and air rushing, the study of the homogeneity and optimization of the impregnation by air rushing. Secondly, the study of the biocidal capacity by plug assays at different times and temperature. And finally, the study of the virus retention in the filter and the RNA damage. Polymeric catalytic filters have been prepared by air rushing and immersion. This is the air rushing setup, which combines the air rush with an automatic table to improve the reproducibility between the samples. The biocidal capacity has been studied through plug assays against different viruses and bacteria. The parameters to be optimized in the air rushing method were the distance between the air rush and the filter and the compressed air pressure. Here I saw some micrographs where we can observe that at short distances the impregnation of the active phase is non-homogeneous, but if we increase this distance the surface is more uniform and there is a better coverage. However, at high distances the impregnation leads to a high agglomeration and worse coverage. So for this reason we select the filters prepared at medium distances and lower pressure to compare with the conventional method of immersion. We can conclude that the air rushing method shows a greater coating with a better biocidal potential. Regarding virus infectivity, we can observe that at room temperature we obtain values below the detection limit except for the rhinovirus, but if we increase slightly the temperature until 37 degrees, we obtain greater results. To study the virus retention, if we observe this graph, we can see that the viral RNA copies are equivalent to the recovery viral titers, so there was no retention in the filter. However, the slight difference between the control with that is the filter without the active phase and the filter with active phase means that there is a partial RNA damage. Finally, the bactericidal activity was evaluated against Escherichia coli and Staphylococcus aureus, and we can see that at room temperature we inactivate Escherichia coli, but we need to increase this temperature until 37 degrees to inactivate the other bacteria. We see that after contact with the catalytic filter there is a a higher dye penetration in the bacteria because of the change in the permeability of the cellular membrane. And finally, the next step of this work is validate these biocidal results in an aerosol chamber which contains aerosols with a bacteriophage. And that's all, and thank you very much for your attention.
Thank you, Alicia.
So this closes this first session. We are way ahead of schedule. You have 10 extra minutes for coffee, but don't be angry if the coffee is still not ready because it's scheduled for 11:30. So you can take this time for voting for the pictures, as I said earlier, and we see each other here at 12 noon. Okay.
Coffee outside.
Coffee's outside, yeah.
So welcome to the next session. And so we get started and we alternate also like in the previous one with the chairs. So the first talk will be given by Merve Keser from the Miguel Alcalde group, Directed Evolution, and she will talk about galactose oxidase engineered by directed evolution, atomistic design, and ancestral sequence reconstruction. Wow.
Thank you.
We'll see. Thank you.
Thank you for the introduction. So my name is Merve. I'm part of the Miguel Alcalde lab. So today I'll be talking to you about the engineered galactose oxidases that we've done by implementing 3 different engineering strategies. So galactose oxidase, the enzyme that I have had comes from Francis Arnold's group, and it has already been engineered to be expressible in E. coli, which previously was not expressed in E. coli, and it had very good expression yields. On top of the expression, also it was engineered within the active site to be glucose sensitive. So the glucose sensitivity that was acquired is the arginine, phenylalanine, and the 406 position that are seen here in these pink sticks. So basically they went from the normal classic galactose oxidase to the aldehyde, and now it's taking glucose into the aldehyde form. So we've taken this already engineered enzyme and applied directed evolution. Directed evolution, some of my colleagues have already said, it basically mimics normal evolution, but in a lab scale, which is way more accelerated, where we apply many different library creation methods. And then we find a host that we can express it for high throughput screening. And then depending on the selection depending on the evolutionary pressure that we set on it, we select mutants, and those hits are then re-put back into the cycle of engineering, expression, and screening. So in my project, we've applied this strategy as well as a computational strategy, which is called PROS. PROS is an algorithm that basically does iterative screening in silico of each amino acid within the scaffold that you put, and basically puts a ranking of the specific amino acid residues that basically confer more stability based on its delta G values. So we've applied this as well, and on top of it, we've also applied ancestral sequence reconstruction, which is basically doing a really extensive multiple sequence alignment in order to find the ancestral or the enzymes that are no longer existing. And the reason for this is because they're generally more promiscuous and they're generally more stable. So by applying all of these three methods separately, we've gotten from our starting point, which is the MRQW, and then we've applied the directed evolution in PROS. We've mixed these two, the variants that we've received from the screenings, and the PROS, and we mix them into our EvoPROS enzyme. So our EvoPROS enzyme now, from the 9, it has now 31 mutations. And separately, we've also applied like I said, the ancestral sequence reconstruction. And in this one, we revived an enzyme that has 151 mutations. This is about 25% of the amino acid composition of this enzyme. Another thing to mention is that this enzyme is very difficult to mutate and engineer because it's just beta sheets, so it's very rigid. So continuing with this strategy, we've had these top 3 variants, and then we said, why not mix all of this together into a final construct named SARLAC. So SARLAC basically uses a scaffold of the ancestral enzyme and integrates the mutations that we received from the EvoPROS variant. So with this, we were able to, so this is a bit of an overview of the engineering process. It's a little convoluted, but it's basically directed evolution, PROS, then we mix directed evolution and PROS together to get EvoPROS, and separately we have the ancestral enzyme, and then we've mixed everything together. So as you can see, the total thermostability improvement is from the starting point. Basically, we ended with a 3.5-fold improvement, and with ancestral was 5.8. So when we mix these two, we have something in between, but still way better than what we started with. And the total activity improvement in regard to galactose and glucose, we see that we still maintain the glucose and the galactose activity, because normally when you engineer an enzyme, there's always a trade-off between activity or stability. However, in our case, luckily, we were able to maintain both the activity of the glucose and the galactose, as well as improving the thermostability. So, of course, we wanted to apply it to catalysis and to transformations. We've selected some benzylic alcohols and very bulky alcohols to see if that we could use some of this stuff for building blocks in green chemistry. So those compounds that are basically in the squares, these two are very valuable for building blocks for pharmaceuticals and diphenylmethanol. This is to check to see how the active site, after such extensive engineering, accommodates such bulky substrate, but also because this is used in UV curing processes. And we were able to see that our enzymes have varying different aspects. And we see that as the chain gets longer, the activity decreases. However, in our final variant, the SARLAC, we do see that it is way more promiscuous with these bulky substrates, and that it is in regard to the MRQW as well as so this is a very thermostable enzyme as well as an ancestral enzyme. So these two are probably our best ones, except that SARLAC now is more promiscuous, therefore we can really kind of more focus on engineering it further. So that was fantastic results, as well as we also got insane thermostability. So between the MRQW, we had 44 degrees. And by our ancestral, it was 71 degrees. So this is about 25 plus degrees increase, which was really fantastic. And as we're going down through the evolutionary, we increase promiscuity as well as thermostability. And when we move this to a Pichia-Pasteur's host, we had really good expression. So the engineering methods that were involved, like I said, were directed evolution, the process-atomistic design, and the ancestral sequence reconstruction. The variants, these are all exclusively, separately mutual, because you can use them for different, because they all have different promiscuities and they all have different thermostability. So depending on what you would like to use it for, you can engineer them further. However, we do believe that our final variant, Sarlacc, is basically in between the engineering campaign that we've run, as well as has really nice expression yields and a very decent thermostability. So this is a very great starting point for further engineering in order to do green chemistry. I'd like to thank my lab mates and of course my advisors and my professor for helping me through this whole PhD project, as well as I was contracted by the Marie Curie program. So this was funded by the Marie Curie Implant Sense program. And I was also contracted by Eon Technologies that also funded my project. So thank you.
So thank you very much. So we go to the next speaker, it's Jesús Laborda Mansilla from Biología Sintética y Biocatalysis Group. He was talking about ancestral sequence reconstruction, direct evolution, complementary strategies for enhancing PET hydrolysis.
Good morning. Well, I'm going to speak about ancestral sequence reconstruction and directed evolution, complementary strategies for enhancing PET hydrolysis. First of all, I would like to introduce you in the huge problem that we have with plastics. And well, I'm going to focus on PET, plastic widely used in bottles and textile fibers. As an alternative to chemical and mechanical recycling, we bet on enzymatic recycling. Well, we are going to use an enzyme, the PTase from Medionella sacchagensis, which is able to break down the PET, breaking the ester bond at 40 degrees. Well, in nature, this enzyme is not optimized, so we want to optimize it to improve the yields to obtain the monomers. Our approach is the following. We started doing ancestral sequence reconstructions, which allows us to obtain ancestral putative enzymes that we are going to express in modern host and introduce in a directed evolution platform. Then we are going to improve the PET hydrolysis activity, and we are going to characterize all the variants. Well, the ancestral sequence reconstruction is based on the use of extant proteins to obtain ancestral sequence putative enzymes that we are introducing in the modern host and checking its activity. We chose one of them and we introduce it in a directed evolution. Well, the key point in this directed evolution cycle is the screening method, which is based in 3 reactions. A fast esterase assay, just to check active mutants. A second one with BHET as a model substrate of PET, which where we are going to check how the reaction is clarified, and the use of nanoparticles, and we are going to measure the products that are released by fluorescence. The last rescreening was confirmed by HPLC. Well, this is the workflow. We started with the ancestral enzyme, produced the library, checked which one has the best activity against PET, and this is the parental for the second generation. After 4 generations, we have our best variant. This is checked by the HPLC, as I said, and we purify all the variants. Now we are going to characterize them, and we can see here how one of the ancestral enzymes had improved the thermostability. After directed evolution, there wasn't any trade-off stability activity between all the variants, and we checked the pH stability, and we had several differences at low pH in between ancestrals, but after directed evolution, we didn't see any change in this trend. And well, finally, concerning the characterization, the activity, well, we have started from an ancestral enzyme here down, and after directed evolution, we have increased the activity, and even more, we have changed the optimal pH where this enzyme works. The native one works better at basic pH, and we have moved it to more neutral pH. So, well, thanks for all, and if you have any question, I will be around here.
So thanks a lot. I see some people in the back standing. We have some places here, so don't be shy and come to the front. I guess nothing happens. So the next speaker is Laura Martinez Quintana from the Green Chemistry and Catalysis group, and she will talk about CO2 methanation over ruthenium supported by a cyanide nitrogen, which is doped with magnesium and barium.
Okay, so thank you for the presentation. Sorry. So as you already know, the increasing concentration of carbon dioxide in the atmosphere due to anthropogenic activity is raising air temperature and causing global warming. That's why great efforts are being made to use renewable energy sources to transform the CO2 into value-added products. In this case, using ruthenium-based catalysts to transform CO2 into methane, which is a valuable energy carrier. So the graphitic carbon nitride, or C3N4, was prepared by thermal polymerization of urea, and then 1% of ruthenium was incorporated by incipient impregnation to give the undoped catalyst. Then doping with the alkali metal was carried out using two different methods. On the one hand, by mixing the alkali and the urea before the thermal polymerization, and then on the other hand, by incorporating the alkali metal and the ruthenium at the same time by co-impregnation. We move on now to the characterization of the prepared catalyst. These are the X-ray diffractograms where we can see the two characteristic peaks of the C3N4. This peak is related to the interlayer stacking of the conjugated system, and this one is related to the implanar structural packing of the 3S-triazine units. Both peaks are maintained in every catalyst, but with the incorporation of the alkali metal, we can see that there is a decrease in crystallinity, and also that the first peak is shifted to lower diffraction angles, which may be due to the incorporation of barium or magnesium in between the layers of the C3N4. And also in the catalyst prepared by co-impregnation, we see the presence of barium carbonate and magnesium oxide that are phases that we don't see in the catalyst prepared by mixing the alkali and the urea before the thermal polymerization. These are the BIT results, and in hydrogen TPR, we observe that the ruthenium support interaction is stronger in the catalyst prepared by co-impregnation. Then in CO2 TPD, we see that the catalyst prepared by mixing the alkali and the urea before the thermal polymerization have more basic size than the catalyst prepared by co-impregnation. In electronic paramagnetic resonance, we obtain this Lorentzian line centered at G2.001, which is attributed to the presence of ampere electrons on carbon atoms within aromatic systems. This signal is more intense in the dope catalyst, which indicates the increase of ampere electrons on the aromatic system and the increase of the presence of nitrogen defect. And this means that the alkali may improve electromobility. And also, the signal increases in the spent catalyst prepared by mixing the alkali and the urea before the thermal polymerization, while the signal decreases in the spent catalyst prepared by co-impregnation. In the nitrogen XPS, we observe the characteristic signs, peaks of the C3M4 with the sp2 nitrogen of the triazine units, the nitrogen of the amino groups, the quaternary nitrogen, and the last peak is from charging effects. In barium XPS of the barium catalyst prepared with 1 millimole of barium, we see the presence of barium carbonate in the catalyst prepared by co-impregnation, as indicated by X-ray diffraction. Also, with the ratios obtained from XPS, it's interesting that the nitrogen-carbon ratio significantly decreases when doping with magnesium, and that may be due to the magnesium that has more influence on the C3M4 structure variation. And when doping with barium, the ruthenium dispersion is higher when doping using the first doping method that was mixing the alkali and the urea before the thermal polymerization. And these are the results of the catalysis performance. We obtain a higher catalytic activity with the barium doped catalyst compared to the undoped one. And also we obtain a higher catalytic activity when doping using the first doping method, mixing the alkali and the urea before the polymerization for both barium and magnesium. And moreover, the methane selectivity was also higher using this first doping method. So finally, the best catalytic activity performance was obtained with the catalyst doped with 1 millimole of barium using the first doping method. And as we have seen, that may be due to the incorporation of barium in the structure to a higher ruthenium dispersion, a lower ruthenium support interaction, and to the presence of more nitrogen defects. And that's all. Thank you for your attention.
Thank you very much. Next presentation will be from Irene Martínez Salazar from the Energía y Química Sostenible group. She was talking about desarrollo y escalado de catalizadores basados en níquel alúmina para la producción de hidrógeno a partir de birreformado de biogás.
Dale a reparar que antes sí que es abierto.
¿Sí?
Sí. Y dale a lo que y ya está. No sé por qué hace eso. Okay, hello everybody. I'm Irene and I'm going to talk about the development and scale up of nickel aluminum oxide based catalyst for hydrogen production from biogas by reforming. So this is part of the Colibri project that I recently joined in June. And the global objective of this project is the use of biogas for the production of hydrogen and its use for new fuel cells. And our objective is the study of new efficient and low-cost catalysts for biogas reforming. So, biogas reforming is an endothermic reaction due to the high chemical stability of methanol and CO2, and therefore it's favored at higher temperatures, which are above 750 degrees. For a catalyst to be able to do the biogas reforming, it needs to have high resistance to sintering and vaporization of the active sites, to poisoning, and to the activation of carbon because of the decomposition of methane at higher temperatures. The catalysts developed in the literature are mainly based on nickel, and the activity and the activation is influenced by size and morphology of the particles, nickel particles size. So far, this reaction has not been implemented at industrial scale. So what our group has been working on is the synthesis of nickel-based catalysts on one side with aluminum oxide supports that are modified by promoters such as lanthanum, cerium, and magnesium, and on the other side by perovskite-based support. And this is all being compared to a commercial nickel catalyst. So the reaction takes place in a fixed bed reactor. And first of all, it suffers a reduction in C2 followed by the reaction at 800 degrees. The parameters that we need to take into account to choose the best catalyst are the conversion of methane the yield of the hydrogen, and also the formation of the carbon. So here we show the conversion of the yields of the different catalysts that were synthesized, and you can see that the ones that show the best activity are the peroxide, one of the catalysts with cerium modified support, and the other with magnesium modified support. And even though this magnesium modified catalyst doesn't have the highest activity, is the one chosen because it has the lower cost. So the next step is to study the stability of this catalyst, and we've done it in that same fixed bed reactor for several cycles. And as you can see here, after 90 hours, we'll still have good stability of the catalyst. So then we move to the next step, which is the scale-up and conformation of this catalyst. What we want to do is to deposit the catalyst on the surface of metallic structures. And we're doing that through wash coating. So we need to optimize these parameters, which are the catalyst suspension and the dip coating process. So basically, the wash coating process involves the immersion of the structure in the catalyst suspension, which needs to have the right viscosity and stability. So when we remove it from the suspension, the catalyst stays on the surface of the structure. So the parameters that we had to optimize are the catalyst particle size, the catalyst concentration, the pH, and also the percentage of surfactants and binders. And regarding the dip coating process, we had to study the immersion and removal speed. So basically, in conclusion, we had a stable nickel-based catalyst that has been selected for the hydrogen production from biogas by reforming. And the method of incorporation of this catalyst by wash coating has been optimized. And for future work, we want to do the synthesis of a second generation catalyst of bimetallic catalyst. Acknowledgements to the rest of my group, Elena, Noelia, and Rufino. And thank you all for your attention.
So thank you. So the next speaker is already there, and is Andrea Menes Rubio from the Directed Evolution Group, and she will talk about the study of recombinant unspecific peroxygenases. Wow, that's a complicated title. Through crystallography and reaction analysis. The rest I don't pronounce because I don't know how to.
That's fine. Okay, I'm Andrea. As she said, I'm working at the Directed Evolution of Enzymes group. And again, I'm going to explain a bit another study with unspecific peroxygenases that is being framed in my thesis. So, as we have heard today so many times, unspecific peroxygenases are enzymes that are for great applications in the biotechnological field because they have because they have an in-group, and with that they can perform so many different selective oxyfunctionalizations of carbon-hydrogen bonds, only using hydrogen peroxide. In my case, I work with some EUPOs that are unusual. They came from an East African incap fungi. And in the lab in the past, Patricia Gomez could functionally express two of them and characterize them. And we have observed that these two enzymes are of particular interest in terms of their properties, like the pH range and the thermostability. Along this work, some of our colleagues from the Institute Blas Cabrera have already crystallized these two enzymes, and they are also performing different studies about protein ligand interactions with these enzymes. And these kind of predictions are what takes me to the main goal of my project, that is the analysis of the oxidative pattern, specifically of this Satyrella UPO2 with a panel of many different substrates that are of industrial interest to search for novel enantiomer ratio and stereoselectivities. So one of the key points of this project was to select the proper substrate. So in this case, we have selected 3 different groups of molecules that are for great application, that are the alkanes, the fatty acids, and the non-isoprenoids. In the case of alkanes, we have chosen dodecane and tetraecane for our studies. The main interest of these molecules is that they possess inert bonds that are a challenge for organic chemistry because it's really hard to achieve oxygenation of these carbons. But UPOs have been reported to do that, so we wanted to explore it with our satyrellas. In the case of fatty acids, they have similar kind of bonds, but also the byproducts of their overoxidations are of huge interest in cosmetic field, for producing biofuels, for producing polymers, resins, and also building blocks. And in the case of non-isoprenoids, they are also of huge interest, mainly in the cosmetic field. So the main workflow of this project was to produce the enzyme Implica pastoris at a bioreactor scale, to purify the enzyme, and then to perform the enzymatic reactions with those different substrates for further analysis by gas chromatography. Also in conjunction with the soaking experiments that have been performed by our colleagues. So I'm not going to enter into much detail about the results, but mainly soaking experiments were consistent with the results we saw with the GC-mass. Okay, so in the case of alkanes, we could achieve subterminal hydroxylation because we can see, for example, alcohols, keto alcohols, and the alcohols. And in this case, dodecane seems to be a better substrate than tetraecane. In the case of fatty acids, it happened more or less the same. We could achieve omega subterminal hydroxylations, and we could observe only alcohols. But soaking experiments suggest that maybe we could achieve also alpha hydroxylation. And right now, with other fatty acid derivatives, we are trying to explore this possibility. And least but not last, with the non-isoprenoids, we could also observe interesting results because we could see hydroxylation not only in the ring itself, but also in the side chain of the molecule. So as a main conclusion of this work, Satyrella UPO2 is a promising biocatalyst for the hydroxylation of alkanes, fatty acids, and non-isoprenoids that are of great interest in the biotechnology field. But the work doesn't stop in there, and we are trying more substrates like the HMF or the FFCA. We are also trying other enzymes, for example, the mutant GROGU, and also we are trying to implement some computational algorithms and also protein engineering techniques that are available in our lab to explore even more the promiscuity of these enzymes. And that's it. You can find my poster outside, and thank you all.
Thank you very much, Andrea. So we go next speaker. Is Laura Molina Nogal from Caracterización Avanzada de Catálisis Heterogénea Group. She was talking about artificial intelligence driving material for solar thermochemical hydrogen generation.
So, okay, I can't. Sorry. So, thank you for the introduction. Good morning, everyone. I'm going to show you the most. Okay, can you hear me well? Okay, I'm going to show you the most relevant aspect of my thesis, supervised by Alicia Bayón and Verónica Ganduglia from ICP CSIC, and from Silvia Jiménez from University of Alcalá de Henares. There are multiple ways to produce hydrogen, but in our research we are focused on thermochemical cycles because they are very promising converting solar energy into hydrogen. They show a theoretical efficiency of 30%, And to reach this, we use perovskites. In the cycle, the perovskite is reduced with heat, and at this point, oxygen vacancies are formed in the structure. So the reduced species splits water and hydrogen is directly produced. The capacity of the perovskite to form oxygen vacancies in the structure is measured by the oxygen vacancy formation enthalpy, whose ideal range must be between the minimum energy for water splitting and under the maximum energy where no thermal reaction occurs. So we have to find the best candidate of the perovskite that has the best combination of B and A elements to keep in this range of enthalpy. So we work with double perovskites, and let's explore all of the possibilities. Firstly, we have to discard all of these unwanted elements to finally have this. And it seems to be easy, but the reality is that we have more than 300,000 materials to explore. So here, in order to avoid thousands of experiments, we need to apply artificial intelligence. And to do this, the methodology has been the following. Firstly, a database search, where we only found 2 theoretical databases. With this data, we've developed a new library of properties that contains DFT calculation and fusion engineering. When the database is ready, we apply the machine learning algorithm to predict the optimal perovskite. In terms of database development, our database contains the formula of the double perovskite, the descriptors, and the target variable, which is the variable to predict. To sum up, the dataset contains double perovskites from different sources, so they are calculated from under different DFT conditions. This means that by the time you apply a machine learning algorithm, you cannot mix the data, and this is one of the most important aspects before applying any machine learning algorithm. So finally, we have 2 machine learning models, where we can see that in the case of Vieten database, we have a higher prediction capacity because we have a higher value of R², and this is because the variability in the double perovskites. But it's true that we also have a higher value of absolute error because the number of entries in this database is less than in this one. So at higher values of oxygen vacancy enthalpy, the prediction is worse. And analyzing this, we thought, okay, we want both features in the database. We want variability and high number of data. So taking into account that the Baldassari computational method is more consistent and her results are easily reproducible, our starting point has been this. So to complete the database, we are recalculating the double perovskite proposed by Vieten using a computational method similar to Baldassari. But the thing is that perovskites are very complex systems, so we have to know how to create the vacancies in the structure. Firstly, we have to calculate the energy in the oxidized and reduced species. So here we have an example of a promising double perovskites, where we have studied the different oxygen in the structure. We studied the nearest neighbors of each oxygen, we remove them, select the most labile ones, and obtain the most stable reduced species, as it happened experimentally. Therefore, the take-home message would be, before applying any machine learning algorithm, we must know our database deeply. If we reach variability among the data, we will improve the accuracy of the prediction of the machine learning algorithm. We have applied an accurate method to generate oxygen vacancies to simulate experimental conditions. And finally, thanks to DFT calculation, we are completing the database and we are reaching variability and high number of data. And thank you for your attention.
So, thanks, Laura.
So next speaker is Elvira Moreno Cortés from the CO2QC group. She was going to talk about photoactive catalysts for CO2 reduction.
Thank you for the introduction. I will be talking in Spanish about what we do in our research group. Bueno, uno de los puntos principales, lo que nos enfocamos nosotros, se trata en reducir el CO2 para transformarlo en productos de valor añadido, y para ello pues seguiremos el esquema así de manera muy sencilla que se muestra en el índice. El primer paso que nosotros tuvimos en cuenta fue sintetizar distintos catalizadores que fueran selectivos hacia estos productos que mencionaba anteriormente. Por cuestiones de tiempo, mencionar brevemente que sintetizamos principalmente estos cuatro, que son el óxido de titanio, el óxido de cobre 1, óxido de tántalo dopado con nitrógeno y el carbonitruro grafítico. De todos ellos, además, para el caso del óxido de titanio, también preparamos composites depositándolos en FTOs mediante electrodeposición electroforética con dos tipos de compuestos. Los primeros de ellos serían nanopartículas de oro que han sido estabilizadas en citrato y en tiol, y luego además composites del óxido de titanio con el óxido de cobre. Una vez que ya tuvimos sintetizados todos estos cuatro catalizadores, además de los composites, lo que hicimos fue caracterizarlos. En primer lugar, hicimos una primera caracterización físico-química en la que de todos ellos obtuvimos fases cristalinas de los correspondientes catalizadores, menos en el caso del óxido de tántalo dopado con nitrógeno, que obtuvimos un amorfo. De todos ellos, destacar que presentan un band gap más o menos dentro de los mismos valores establecidos, pero que por otro lado sus propiedades texturales varían mucho entre ellos. Además de la caracterización físico-química, también nos centramos en la electroquímica. En primer lugar, hicimos dos. La primera de ellas se trata de la voltamperometría cíclica. Pero para quien no lo sepa qué es la voltamperometría cíclica, es un proceso que sirve para ver pues, los distintos procesos redox que tienen lugar en nuestro sistema de análisis. Caracterizando con las condiciones que se muestran aquí, lo hicimos para los cuatro catalizadores principales. En todos estos gráficos se muestra en rojo el catalizador cuando fue irradiado por la lámpara de xenón y en negro aquellas que corresponden al catalizador sin irradiar. En todas ellas podemos observar, tanto en la parte de arriba del gráfico, todos los picos de oxidación, mientras que abajo corresponden a los de reducción. Destacar que para todos los catalizadores, salvo para el óxido de tántalo dopado con nitrógeno, todos presentan una mayor fotocorriente inducida para un rango del potencial aplicado, es decir, son mucho más fotoactivos cuando se les irradia la luz procedente de la lámpara. Además también de la voltamperometría cíclica tenemos la cronoamperometría, que se basa en determinar la fotocorriente inducida en los electrodos, mientras que se van alternando periodos de irradiación y de oscuridad. Las condiciones son exactamente iguales que utilizamos para el caso anterior y en este caso me gustaría destacar el contraste que existe entre el óxido de titanio y el óxido de cobre 1. Como podemos comprobar en este primer caso, lo que estudiamos a diferencia del caso anterior es la intensidad de corriente frente al tiempo. En el caso del óxido de titanio, esta intensidad de corriente, alternando cada 20 segundos la presencia y la ausencia de la luz procedente de la lámpara, se aprecia que es mucho más estable a lo largo del tiempo, al contrario que en el caso del óxido de cobre, que va decayendo, lo que podemos atribuir a distintos procesos de oxidación que tengan lugar en el proceso. Una vez que ya tenemos todos los catalizadores sintetizados, procedimos a reducir el dióxido de carbono. Para esto utilizamos dos vías distintas. La primera de ellas es la fotorreducción del CO2. Para ello pues, se preparan distintos tubos de reacción con las condiciones que se muestran en la tabla. Se burbujea CO2 durante 30 minutos y luego se introducen en el sistema de reacción que podéis ver en la foto. Nosotros con lo que contamos en este reactor es con una lámpara de mercurio que se mueve dentro del rango del ultravioleta C y en su interior pues colocamos los distintos tubos alrededor de la lámpara. E realizamos distintos ensayos a distintos tiempos, como podéis ver aquí en el gráfico de barra, y se realizaron en 0, 2, 4, 8 y 24 horas. En todos ellos usamos también metanol y etanol como otros productos, pero el mayoritario en todos los casos fue el monóxido de carbono, de ahí a presentar en este caso. Como podemos observar bien en el gráfico de barras y según se muestra aquí en el ejemplo de la tabla de la derecha con el gráfico de velocidad, podemos observar cómo existe un pico máximo de producción del monóxido de carbono en todos los casos que corresponde a las 8 horas de reacción. Por lo tanto, podríamos estimar que en este caso, para estos catalizadores, para este tipo de reacción, sería el tiempo óptimo. En cuanto a la proporción y la obtención del monóxido de carbono, esta conversión iría en el caso de, en orden creciente, primero tendríamos el óxido de titanio para la conversión a monóxido de carbono, pasando por el carbonitruro grafítico, para seguir luego con el óxido de cobre y el óxido de tántalo dopado con nitrógeno. Este sería uno de los primeros procesos. Luego contamos también con la fotoelectroreducción del CO2. Para ello utilizamos la celda electrolítica que se ve aquí en la imagen, utilizando pues, unos FTOs como electrodo de trabajo. Las condiciones óptimas de deposición que obtuvimos para todos los catalizadores y los composites son las que se encuentran aquí en la tabla.
Entonces, en este caso lo que hacíamos es utilizar un potencial elegido mediante estudios previos de 0,5 voltios y burbujeando continuamente nitrógeno. Teniendo en cuenta ello, obtuvimos los siguientes resultados. Se muestra en la tabla un estudio analizado para las 4 horas de reacción, en la que los compuestos principales para ellos son el monóxido de carbono, el ácido fórmico y el metano. Entonces, aún quedarían muchos más estudios pendientes para establecer y buscar otros distintos tipos de franjas, porque en este caso solo completamos hasta las 4 horas, pero es algo que podríamos llevar a cabo en futuros análisis. Por último, me gustaría agradecer a los proyectos que nos financian, a la unidad de apoyo, a mi grupo de investigación y a vosotros por vuestra atención.
So, thank you, Elvira, for the talk. So the next speaker will be Eric Moreta Urbano from the Synthetic Biology and Biocatalysis group, and he will talk about synthetic yeast for PET degradation.
Okay.
Please.
Thank you for the presentation. So I'm going to talk to you about the work I did in the high intro IQ scholarship I did during 4 months in the Synthetic Biology and Biocatalysis group. So using synthetic yeast for polyethylene terephthalate degradation, or PET. So I want to start by describing to you what are synthetic yeast. So basically, a synthetic yeast is a synthetic yeast whose chromosomic DNA has been replaced with a synthetic version, which has been designed with some criteria in order to give it more genetic stability. Some of these modifications are to get rid of introns in the genes, getting rid of transposons, defining some PCR tags, and defining some LOX-P sites in order to do further experiments. So obviously, this is easier said than done. That is why a worldwide consortium of labs working on synthesizing each chromosome and then stacking them up in one cell by sexually crossing them. And in the work I did here, I used 2 synthetic strains. One which I'm going to refer as synthetic 2, which has the synthetic chromosome 2, and a synthetic yeast that has 3.5 synthetic chromosomes, which are the chromosome 2, the chromosome 3, the chromosome 6, and the right arm of the chromosome 9. What we are focusing on the PET problem. So as you already know and heard, we produce a lot of plastic. Just plastic packaging accounts for the 70% of the whole market of consumer products, and just PET accounts for the 12% of the global solid waste. And we have 3 ways to treat and degrade PET, which are the mechanical, the chemical, and the biological waste, being the latter the one I am focusing on. I'm going to talk to you a little bit of the project in general. So first, in the lab, they took the sequence of the PETase of Idonella sakaiensis, which is a bacteria that degrades PET. Then in the lab, they performed ancestral PETase reconstruction and directed evolution, where they got 2 versions of the PETase that were promising and worked in mild conditions, which are the PETase-10 and the PETase-73. And the work I did was to make 4 different strains of yeast to produce and secrete these enzymes, and to test how well these enzymes were expressed in each strain. So the results I got: first, I tested the supernatant of the cells with a substrate called paranitrophenylbutyrate. This is a colorimetric assay that allows me to measure how the activity in the supernatant is doing at degrading paranitrophenylbutyrate, and I can calculate the activity of each supernatant. So here you have to take into account that the activity does not relate to the enzyme itself, but how well the strain produces and secretes the enzyme. So as you can see in the chart and in the graphic, here you can see that the wild types are which are the BJ and BY. These wild types are producing the enzyme and are degrading the paranitrophenylbutyrate, but the synthetic yeast, the synthetic 2 and synthetic 3.5, almost do not produce the enzyme. Especially the synthetic 3.5, we see no activity at all in the supernatant. But we also wanted to test it in another substrate, which is BHT, which is a chemical species that is a byproduct of the PET degradation. thing about the BHT is that it is not soluble in aqueous solution, but when it is furtherly degraded by the PETase, it forms other species that are soluble, and we can measure the degradation of BHT looking at how the turbidity of the reaction goes down along the time. So having this into account, we can see that the wild type strains, which are the BJ and BY, do decrease the turbidity, which means that are degrading the BHT, and the synthetic 2 do show some degradation of the BHT, but the synthetic yeast 3.5 just do not degrade the BHT. Finally, we wanted to test in another substrate, which is nanopet, nanoparticles of PET, and we wanted to measure the chemical species that are formed when PET is degraded, which are MHT and TPA. This was measured by HPLC, and here you can see a chart how the reaction evolves over time. So as more evolve the reaction, you get more TPA than MHT. Having this in mind, you can see that again, the wild type yeast secrete better the enzyme, the reaction goes faster. Synthetic 2 strain produce the enzyme but do not go that much faster, and the synthetic yeast 3.5 just do not show any activity at all. So as conclusions, the synthetic yeast show less PETase expression than wild type yeast, yet we can use these cells and improve them by induced evolution due to their genetic characteristics. And I want to thank you for your attention, to the collaborators and to the funding agency.
Thank you very much. So we go to the next speaker is Julia Murciano Brea from Directive Evolution of Enzyme Lab. She was going to talk about degradation of bioplastic through schema RASP, homogeneous recombination, and ancestral sequence reconstruction.
¿Se me oye? Sí. Okay, hello, I'm Julia Murciano. Thank you for the introduction. And I'm gonna talk again about plastics. You know a lot about the context because it has already been discussed today. But the main problem with plastics is their mismanagement at their end of life. For they finish being accumulated into landfills or being incinerated, and to a much lower rate being recycled. Some solutions that come inspired by this threat is to create new materials such as bioplastics. Some of them have already been discussed today, like polybutylene succinate, and they are indeed greener than traditional plastics, but have their problems, because if they are not properly managed at their end of life, they can also be ending up in landfills. So the other solution for this type of problem is the biodegradation. In this case, I'm going to present the enzymatic biodegradation of plastics, highlighting cotinases as a great alternative, for they have wide range of substrates and have already been proven to be active against multiple plastics. The thing with cotinases is that they are active normally against one or two plastics and are not active against multiple plastics. But in a recycling setting, we are looking towards an enzyme that could degrade multiple plastics. For this, some work that has been done in the laboratory by Diana Toledo, who will talk later today, during the Revolution project, she took 3 different parental enzymes that had cotinases that had activity against different plastics, mixed them up together using an algorithm called schema RASP, that is a computational algorithm that makes chimeras by choosing the best crossover points in the cotinases and rearranging the blocks to create new enzymes that have supposedly the best parts of the initial cotinases. So she created a lot of libraries, 200 libraries, and chose one with 2,000 clones, in which 14 of those were screened and selected, and 12 of those chimeras were highly active against plastics, which were great news. Following her steps during the project I'm now part of, which is the Blade to Circ, some of those chimeras are now being improved even further by the means of directed evolution. This has already been discussed many times today, so I'm not going to go too much into it, just to mention that the screening we are doing is maybe the most different part, because we are using the final target
which is the substrate, the PBS is a plastic, in a suspension in water, which allows us to measure in kinetic mode how the plastic is being biodegraded by the cutinases. In a question of minutes, we can see which mutants are active against our plastics and which aren't, which gives us great high quality data that can be further used into things like machine learning, where we plan to train some models with this highly quality data that will maybe allow us to predict improved variants to screen again. This, of course, at the beginning is a lot of effort, for a big amount of data is needed, but with time it will be very rewarding. That is all, and thank you very much.
So thanks, Julia, for your nice talk. So the next speaker will be Verónica Najarro Ovejero from the Green Chemistry and Catalysis group, and she would talk about induction heating applied to anisole HCO using formic acid as hydrogen source.
Okay. Gracias por la presentación. Como ya han comentado, me llamo Verónica y voy a comenzar. Actualmente las biorefinerías suponen un método más sostenible en comparación con las refinerías clásicas de combustibles fósiles, ya que aprovechan y tratan restos de biomasa de sus componentes para generar compuestos que se utilizan en la industria química y para producir combustibles. Entre ellos destaca la biomasa lignocelulósica, compuesta por hemicelulosa, celulosa y lignina. La lignina es una de las partes la más interesante, la verdad, ya que está compuesta por cadenas poliméricas de aromáticos. Estas cadenas son tratadas con procesos como la pirólisis para obtener moléculas de menor peso molecular, pero que tienen un alto contenido de oxígeno. Por lo tanto, es necesario llevar a cabo un siguiente tratamiento, que es la HDO, la hidrodeoxigenación, donde se les aplica hidrógeno a estas moléculas para optimizar sus propiedades. Para nuestro trabajo hemos seleccionado el anisol como molécula modelo para estudiar su conversión y selectividad hacia benceno y fenol. Como comentaba, las biorefinerías necesitan bastante hidrógeno para llevar a cabo estos procesos, por lo que hay otras formas de mejorar la sostenibilidad de estos, que es mediante el aprovechamiento de moléculas más pequeñas que se obtienen como subproductos en los procesos principales de tratamiento de biomasa. Es el caso del ácido fórmico, que es una molécula más pequeña, estable y no tóxica, que descompone en hidrógeno y CO2. También hay otra forma de mejorar bastante, de intentar buscar formas de mejorar la eficiencia y la sostenibilidad de estos procesos, que es mediante la aplicación de la catálisis por inducción magnética, que se basa en el principio de que los materiales ferromagnéticos generan calor por pérdida de histéresis cuando se les aplica un campo alterno de alta frecuencia. Por lo tanto, se nos produce un calor de dentro del material hacia afuera, lo que tiene como ventaja producir un calor más selectivo, más controlado y una mayor rapidez de calentamiento, lo que nos conlleva una mayor eficiencia energética. Así que para este trabajo, primeramente se ha llevado a cabo la síntesis de catalizadores de reño, hechos por impregnación en un grafito de alta superficie, que ha sido tratado térmicamente en helio y en hidrógeno para obtener tanto reño metálico como el óxido soportado, seguida de una segunda síntesis de nanomateriales magnéticos, utilizando como precursores hierro y cobalto, para generar unas nanopartículas magnéticas que además han sido encapsuladas en carbono utilizando glucosa. ¿Por qué? Porque este encapsulamiento lo que va a ayudar es a mantener estables estas propiedades magnéticas y van a evitar que estos materiales intervengan en la reacción principal del catalizador de reño. Por lo tanto, se va a llevar a cabo una reacción en fase de gas incontinuo, donde va a quedar un lecho catalítico en el que se combine catalizador y nanopartículas magnéticas. Hablando así un poco de la caracterización, en primer lugar, se llevó a cabo una comparativa entre reño metálico y óxido de reño, donde por XPS sobre todo se vio y se observó que el reño en fase metálica se encuentra más disperso que el óxido de reño. También se llevó a cabo la caracterización morfológica de los nanomateriales magnéticos, donde en primer lugar por microscopía confirmamos que se produce el encapsulamiento para los tres tipos de materiales. Por XRD y por XPS vimos que el hierro y el cobalto se encuentran en forma metálica y que en la síntesis donde se han utilizado los dos precursores se forma una aleación de hierro y de cobalto. Y todos estos materiales tienen un tamaño de partícula medio en torno a los 45 nanómetros. También se llevó a cabo la caracterización de sus propiedades magnéticas hechas por magnetometría, Se compararon los resultados con una magnetita comercial sin encapsular y se obtuvo como mejor material de los tres el formado por la aleación, ya que es el que presenta mayor magnetización de saturación y una menor coercitividad. La coercitividad es la resistencia de estos materiales a desmagnetizarse. Finalmente, en lo que lleva a la parte catalítica, como he comentado, se llevó a cabo la hidrogenación de anisol con ácido fórmico. A 320 grados se seleccionó como catalizador el óxido de renio, ya que se hicieron unos estudios previos comparando óxido y renio metálico y como los resultados eran similares, se decidió continuar con el óxido. Lo que se comparó fue la fuente de calentamiento utilizada, comparando varios materiales magnéticos para contrastar qué campos era necesario aplicar para llegar a la temperatura de reacción. Son campos que varían entre los 15 y los 35 militeslas en función del material y como vemos fue el material formado por la aleación de hierro y cobalto el que necesitó de un menor campo magnético. En lo que ya refiere a los datos de conversión, se mantiene estable en todos los casos, para todos los materiales. Hay una desactivación del catalizador al principio que está relacionada con el descenso en la selectividad hacia el benceno, lo que viene a la par con un aumento en la selectividad hacia el fenol y el tolueno, que no se muestra. Pero el que la estabilidad de la conversión y el que no varíe al cambiar los materiales magnéticos de una reacción a otra, nos indica que este tipo de encapsulamiento con carbono es efectivo. Y eso es todo, muchas gracias.
Thank you very much. Now we go next. The speaker is Karen Cecilia Pájaro Ávila from Catalizadores Nanoestructurados Group. She was talking about influence of the pretreatment on the activity and regenerability of catalysts for dry reforming of methane.
Hello everybody, and thank you for the presentation. Dry reforming of methane has attracted interest from environmental and industrial perspective, since it uses two of the most harmful greenhouse gases, methane and dioxide of carbon, and provides a mechanism to convert them into value products as syngas, that is a mixture between hydrogen and monoxide of carbon. Furthermore, it produces a ratio about 1 that can be used for the feature trough process and oxygenase and also alcohols products. For that, however, the quick deactivation of the catalyst limits his implementation and scaling out of the process, and it's important for that search and new engineering solutions and catalyst design. In this perspective, is important to the end objective of our work is obtain a catalyst nickel seria, improve the reactions for obtain best activity and performance. Our catalysts were prepared by co-precipitation method using two microemulsions, an acid one with a precursor salt dissolved and a basic one to achieve a greater homogeneity and surface area. And the cationic ratio nickel support that we used was 2A. So we characterized the catalyst. We obtained that all catalysts had nitrogen absorption desorption isotherms time 4, characteristic of the mesopore solids that we can observe in the table, and the A2 hysteresis characteristic of the bottleneck porous, and also can be attributed for non-defined distribution of pore size. The surface area of nickel seria increased with the incorporation of samarium and zirconium in the structure. And for the other hand, the LDRX result show that reflection of cerium oxide with a fluoride type structure with no separation to the other phases except for the oxide of nickel in some case. The change that we can obtain in the lattice parameter is for the difference that we can see in the ionic radius of the samarium zirconium when we incorporate in the lattice parameter of ceria, forming the mixed oxide. SPS show from quantitative analysis data that the all catalysts with zirconium obtain enrichment in seria in surface. The addition of samario increases slightly the grade of reduction of seria, and the quantification of nickel indicate that significantly part of them could be in the structure in non-surface state, except for the nickel seria. The result of catalyst tests, we show a continuity. We obtained that the nickel seria had a good activity, but with the time start to decrease. For that, we can think that it could be to the side reaction. Nickel serosamario had a quick deactivation, and the catalyst with zirconium were more stable than the others one. The yield of hydrogen and CO had the same trend of behavior than the conversion of methane and dioxide of carbon, and for the hydrogen CO ratio, show except for the nickel seria samario that all catalysts was about 1.3. For side reaction, in our case, water gas shift. In the case of the nickel seria samario, could be for the side reaction that leads to the formation of carbon and reverse water gas shift. After regeneration, the catalyst didn't recover their initial activity. For that, for example, the nickel seria, it continued with the deactivation observed in the first one. The nickel serio samario didn't recover the activity, and the catalyst with zirconia were stable in the time. In principle, with the nickel serio samario zirconium. That indicates that the deactivation is not only for the deposit of carbon, but also for the modification of the structure, in this case for the sintering of the catalyst that we can observe in the SEM, TPO, and SRD that we made for the post-characterization in the catalyst. So the aim conclusion that we obtain is that the interaction of nickel with the support is the critical factor that determines its catalytic properties, and the zirconia could be or plays a role principal in that activity. Thank you so much for everything.
Thank you, Karen, for my talk. So the next speaker is Cristina Peinado-Cebrián from the Energy and Sustainable Chemistry Group. She will talk about sorption enhanced DNA synthesis for renewable sources using silicotan-rich acid as the acidic catalyst.
Thank you for the introduction. This work was presented at the CSIC Congress in Bilbao last July, which I announced you that will be held in Madrid in the next edition. As an introduction, dimethyl ether, or DME, is a clean alternative fuel that can be used for transport and domestic purposes. But in order to reduce the CO2 emissions related to the use of dimethyl ether, it is mandatory to shift its procedence from fossil to renewable sources such as biomass. In this work, we have studied the alternative one-step DME synthesis process, which, unlike the traditional process in two steps, in which we have syngas that goes to methanol and methanol to DME. In this case, we have the synthesis of methanol and DME both in the same reactor. One of the main challenges for the development of this process is that there is a lot of water inside the reactor, and this effect intensifies when we part from a biomass-derived syngas because its content in CO2 is much higher. This makes this reaction to happen backwards, which is the reverse water gas reaction, producing an extra amount of water. This water is detrimental for the typical materials in the catalytic beds used for the study of this process. So in this work, we have tried to tackle these problems from two different approaches. One of them was to study the effect of the acidic function by comparing two different catalytic beds, consisting in a commercial CCA for the methanol synthesis and either as acidic function, a gamma alumina, or a heteropolyacid based catalyst. In this case, it was a titanium-supported silicotonsic acid, which have already presented very good activity for the methanol dehydration to DME without water deactivation. The other approach was to use the very same catalytic beds, but adding to them a zeolite 3A as a sorbent material to remove water from the reacting medium. And by doing so, we would be shifting the equilibrium towards the formation of more methanol and more dimethyl ether. This process with water sorption can be found in literature as sorption-enhanced dimethyl ether synthesis, or SEDMES. So moving ahead to the results section, I show you here the DME productivity for both processes and both catalytic beds. Let's look first at the direct DME synthesis, meaning without water sorption. And from these results, we can see that the heteropolyacid containing bed was much more active than the gamma alumina containing bed. And this observation was also made at other operating conditions. So we can say that the heteropolyacid containing bed is much more active than the gamma alumina containing bed. When we compare both processes, SEDMES and direct synthesis of DME, meaning with and without water sorption, we can say that the SEDMES process is capable to improving the performance of both catalytic beds, but this enhancement was much higher for the gamma alumina containing bed than for the heteropolyacid containing bed. Where did this difference came from? We calculated this value that can be understood as a methanol conversion for both processes and both catalytic beds. And we observed a huge increase for this value for the gamma alumina containing bed, but not for the heteropolyacid containing bed because there wasn't so much room for improvement already. So this meant that by removing water, we are preventing the gamma alumina to deactivate by water. But this enhancement alone cannot be accountable for all of this increase in DME productivity. So we also calculated the total carbon conversion, understood as the CO and CO2 transformed into methanol or DME, for both processes and the gamma alumina containing bed. We observed a net increase in this total carbon conversion, and this meant that although we were favoring the reverse water Keser reaction by removing water, that can be observed here with a conversion of CO2 negative, meaning that we're producing CO, we were also increasing the methanol formation rate through CO2 hydrogenation. So this enhancement in combination with this other enhancement can explain this huge increase in DME productivity for the gamma alumina containing bed. So as conclusions, we can say that DME productivity in the direct synthesis of DME is higher when using an heteropolyacid function than a gamma alumina function. And we can also say that SEDMES can increase up to 11 times the DME productivity obtained in the direct synthesis of DME. So thank you very much for your attention.
Well, thank you very much. So now next presentation will be from Raúl Peño Mateos. He will going to talk about optimization of reactor operation for hydrogen production via thermochemical cycle.
¿Se me oye? Okay. So, good morning, everybody. Thank you for the introduction. One of the most important things to do when designing a solar plant is the system modeling. Specifically, we want to design the reactor part. For example, the reactor could hold a thermochemical water splitting cycle like that, which is a two-step cycle. that uses a metal oxide, which is non-stoichiometric, to reduce the water molecule and produce hydrogen. The first step, we have the thermal reduction, which is an endothermic reaction favored at high temperatures and low partial pressure of oxygen. Because of that, we need the use of an inert gas in order to favor the reaction to the products. The second step will be the reoxidation, when we can use the vacancies created during the thermal reduction in order to produce the hydrogen. And this is an exothermic reaction which is favored at low temperatures and low partial pressure of hydrogen. And also here we will be using a water current which will be in excess. Because of that, our main objective here is to make an analysis of the main gases, the inert gas during the reduction and the water during the reoxidation, in order to not underestimate the production cost of the hydrogen. We have studied two different configurations. Firstly, the fixed bed, which requires an alternating between the gases and the solar radiation system, which is not that optimal. And on the other hand, we have the countercurrent reactor, which uses two separated reactors for each reaction condition. The main advantage here is that we can make the particles move around the chambers, which makes our chemical potential higher. How did we resolve our reactors? We make some simulations using MATLAB, and for the fixed bed system, we resolved them by iterative numerical methods, which were native in the software. And for the countercurrent system, we assumed chemical equilibrium along all the flow path. We take into account some common conditions like the temperature, the material, and the kinetics. And also, it's important to remark that we used the lambda term to track the consumption and the non-stoichiometric coefficient to track the reaction process. When passing to resolving those systems, firstly the fixed bed, we can see how the main parameters affected the process, and also we came to the idea that it is not that desirable to achieve the chemical equilibrium because we could save some inert gas in the process. When comparing the fixed bed and the countercurrent reactions, we saw how the countercurrent outperformed the fixed bed. This is because for the same extent of reductions, which means the same reaction point, we could acquire that by using less inert gas. When we could make also a techno-economic analysis, we saw that the lambda term is, well, the cost of the production of hydrogen is highly dependent of that lambda values. And we fixed a threshold where lambda values between 30 would imply a cost of hydrogen over $1, below $1 kilo. And that work allowed us to make some conclusion, like the main parameters that affected the process, that it is not that desirable to achieve the chemical equilibrium, how the countercurrent outperformed the fixed bed, and the values of lambda greater than 30 will increase the cost of hydrogen production above $1 kilo. And that's all, and thank you for your attention.
So, thank you. The next speaker is David Rodriguez Garcia from the Applied Biocatalysis, and he would talk about glycoside and acyl glycosides through synthesis through enzyme catalyzed reactions. Yes.
Yeah. Good morning, everyone. My name is David Rodriguez, and I'm going to talk about the topic glycoside and acyl glycoside synthesis of the flavonoid ampelopsin through enzyme catalyzed reactions. And this work belongs to my master thesis. Polyphenols are natural products that can be found in plants such as vines and olive trees. And among these polyphenols, flavonoids stand out because of the antioxidant activity and anti-cancer properties. My study has focused in ampelopsin that can be found in Ampelopsis crossdentata, and this molecule has antioxidant activity, anti-cancer, and antimicrobial properties. However, it has low bioavailability, poor solubility, and low cell membrane permeability, which limits its applications. So the objective was the synthesis of glycosides and acyl glycosides of ampelopsin through biocatalysis, and further measurement of activity and solubility. For the phase reaction, we use as saccharose phosphorylase. It is a mutant that has been modified to accept large molecules in its pocket. And the second reaction is carried by a commercial lipase. The alpha glycosylation, for this reaction, we carry out several experiments to fit the reaction conditions, such as the amount of sucrose, and the reaction leads to two products. Through mass spectroscopy and NMR, we determine the structure of these two products, and the main product has the glucose added in the 4th position. The second reaction, the acylation, used the major product from the previous reaction, and also we performed 3 different long chain lengths, and these reactions are selective and finish in less than 1 hour. With these 5 new compounds, I did several tests for antioxidant activity. I used 2 different radicals, ABTS and APPH. For the ABTS test, we observed that in general all the compounds have a good antioxidant activity, but for the DPPH test, the major product and the acylated, the antioxidant activity decreased. In the solubility test in water, it is remarkable the minor product that has around 650 times it's more soluble than the ampelopsin. And regarding the gastrointestinal absorption test, both ampelopsin and the products were below the low permeability control. So in conclusion, alpha glycosylation and subsequent acylation of the flavonoid ampelopsin with high conversion were achieved. The enzymes led to selective products, short-time reaction, and inexpensive reagents. NMR and mass spectroscopy were used to determine the structure of 5 new products, and in general, the products show good antioxidant activity and optimal solubility. And glycosylation and acylation of ampelopsin could become a strategy for the synthesis of products that lead to active principles. Thank you for your attention.
Thank you very much. So now next speaker is David Rodriguez López from EQS Group, and he was talking about exploring the horizon of lanthanum nickel oxide-based modified materials, potential bifunctional electrocatalysis in alkaline media.
¿Se me oye? Bien. Bueno, lo primero, muchas gracias a todos. Soy David Rodríguez también. Y bueno, voy a contaros mis últimos avances en torno al niquelato de lantano como electrocatalizador para electrólisis en medio alcalí. Bien, primero un poquito de contexto. Creo que son cosas que todos ya sabemos, pero sí me gustaría remarcar algunas, simplemente. Sabemos todos la crisis que hay actualmente con el hidrógeno, con la falta de energía, la contaminación. Y mucha gente está proponiendo diferentes aproximaciones para encontrar algo que pueda, digamos, soslayar este tipo de inconvenientes. Nosotros hemos propuesto, y mucha gente, el hidrógeno verde, porque es un vector energético y tiene una gran densidad energética. Además, para producir este hidrógeno hemos propuesto la alternativa de la electrólisis. Necesitamos para ello, por tanto, electrocatalizadores. Estos electrocatalizadores van a ser perovskitas debido a su alto grado de modificación, digamos que nos permiten diferentes posibilidades. Y además tienen la capacidad de ser bifuncionales, es decir, van a ser activos para la reacción de evolución de oxígeno y la reacción de reducción de oxígeno, que explicaremos más adelante un poquito lo que es. Pero antes tengo una pregunta para vosotros. ¿Dónde están estos lugares? ¿Alguno tiene idea? Quizá el primero. O sea, se nos ocurre en países lejanos, Rusia, no sé dónde. Efectivamente, Siberia, Rusia. ¿Pero esta? ¿Alguno? Bueno, pues está en Badajoz. No sé vosotros, pero a mí no me gustaría que esto estuviera en Cuenca, en mi caso. ¿Por qué os enseño esto? El elemento mayoritario en la catálisis en medio alcalino para la producción de hidrógeno, o uno de los más importantes, y en nuestro caso también, es el níquel. Estos son minas de níquel. Hay que tratar de evitar utilizar tanto níquel en nuestros materiales. Así que con esto en mente nos hemos embarcado en un proceso iterativo de mejora para tratar de reducir el contenido de níquel a partir del niquelato de lantano. Inicialmente dopando con molibdeno, aumentando el área de ese material y modificando su morfología. Voy a hablaros un poquito de las formas de síntesis, pero tampoco me quiero detener en eso. Simplemente deciros que inicialmente hicimos un enfoque mecanoquímico para las estructuras más sencillas como el niquelato de lantano base. También nos sirvió para doparlo con molibdeno y además en cuanto al dopaje de molibdeno pudimos comprobar que el grado de dopaje específico de molibdeno que para nosotros mejor funcionaba era el del 10%, dado que otra cantidad como el 15 o el 20% generaba una cantidad de impurezas que digamos el grado de el compromiso entre la cantidad de impurezas y la actividad ya no nos parecía tan óptimo. Para continuar, aumentamos el área añadiendo tilosa, y esto fue una modificación del proceso mecanoquímico anterior en el cual le tuvimos que hacer un tratamiento previo. Básicamente era tratarlo con un molino de bolas y después pudimos comprobar, de nuevo mediante difracción de rayos X, que conseguimos esa estructura no sin una pequeña cantidad de impurezas de óxido de níquel y otras fases de lantano molibdeno. Por último, la cuarta de nuestras estructuras fue aquella que tenía una morfología diferente, si bien conservaba la estructura cristalina de perovskita. Bien, aquí nos valimos del proceso solvotermal para llevarla a cabo. Sí quiero detenerme en la caracterización de estas nanosferas. Utilizamos sobre todo para ello la técnica de SEM, y pudimos ver que efectivamente teníamos la proporción adecuada de elementos que nosotros deseábamos en la estructura. También vimos que efectivamente la morfología, las nanosferas huecas, estaban ahí, como podemos ver. ¿Veis que efectivamente son huecas? Pude encontrar una que estaba partida. Y además pudimos comprobar que la distribución elemental era bastante uniforme a lo largo de toda la estructura, por mapeo en SEM. También quería incidir en que la síntesis de esta estructura y que al mismo tiempo conservara su estructura cristalina, mientras que la morfología seguía siendo la de una nanosfera hueca, fue bastante complicada, y la temperatura y tiempo de compromiso que nosotros encontramos para sintetizarlo fue esta, después de hacer varios intentos. Sin embargo, al subirla destruíamos la estructura. Una vez que tuvimos todos los materiales, nos detuvimos a medirlos en el siguiente setting que tenemos aquí. Hicimos varias pruebas electroquímicas. En primer lugar, medimos la FSA, la superficie electroactiva de el área electroactiva de este material. Inicialmente teníamos en torno a unos 10 metros cuadrados, puede que incluso menos, y después de hacer varias modificaciones llegamos a multiplicar prácticamente por 5 ese área. Esto es muy importante porque la electrocatálisis, como muchas otras, es un fenómeno superficial. Actualmente seguimos trabajando en esta optimización. También podemos medir la reacción de reducción de oxígeno, porque si bien nosotros lo que queremos es producir hidrógeno, la reacción limitante en esa reacción global de la electrólisis del agua no es la de producir el hidrógeno, es la de producir el oxígeno. Entonces nos interesa tanto, en este caso para ser bifuncional, la reducción del oxígeno como también la evolución de ese oxígeno para que este proceso sea lo más rápido y eficiente posible. Podemos ver que con las diferentes modificaciones, casi en todas, hemos llegado a conseguir una mejora de la actividad en la reducción, Y en la evolución, actualmente nos quedaría medir las últimas, las nanosferas, pero hasta hoy hemos conseguido mejorar la actividad siempre que hemos sintetizado un nuevo material. También nos detuvimos a estudiar la estabilidad de este material, dado que nada serviría que fuera muy activo si bien poco estable, lo cual suele ocurrir. En nuestro caso no tanto, hicimos unos 4000 ciclos y la pérdida de actividad, si bien se dio ligeramente, no era algo que fuera realmente alarmante como podemos ver aquí, y sobre todo si comparamos entre la muestra inicial y la muestra dopada con molibdeno, no tenemos una pérdida significativamente mayor entre una y otra respecto a la actividad. Una vez con ello en mente, decidimos escalar el proceso. Escalar el proceso no todavía a nuestro, digamos, objetivo final, que sería el electrolizador, en nuestro caso, uno de esos objetivos, sino a una prueba en placas de laboratorio, que sería una tentativa de ese electrolizador. Y obtuvimos unos valores cercanos a los mejores materiales que tenemos en bibliografía, es decir, 2 voltios a 2 amperios centímetro cuadrado. Por último, contaros un poco nuestras perspectivas de futuro y deciros que, dado que la técnica de cambiar la morfología parece que está funcionando bien, queremos experimentar con otro tipo de morfologías en las cuales estoy trabajando actualmente y continuar con el escalado hasta el electrolizador final. Por último, como conclusiones, deciros que hemos determinado la cantidad óptima de molibdeno como dopaje en esta estructura para conseguir una actividad óptima en cuanto a la bifuncionalidad. También deciros que hemos preparado por primera vez, o hasta donde nosotros sabemos por primera vez, la estructura de nanosferas huecas del niquelato de lantano dopado con molibdeno. Hemos incrementado el área unas 5 veces con respecto a la muestra inicial y además hemos mejorado la actividad con respecto a cada modificación con respecto al original. Eso es todo y muchísimas gracias.
Thanks, David, for the talk. And the next speaker is Paula Sanz Rodriguez from the Directed Evolution Group. And she will talk about computational design of unspecified peroxygenases with FunClip.
So, yeah, you can hear me well. So, thank you for the presentation. Sorry, we have another unspecific peroxygenase, but I work with that in the lab. So, a little bit of introduction. I don't want to say many things because some of my colleagues have talked about and you may have like a little bit idea of what are UPOs. But what important thing is that they have a self-sufficient monoperoxygenase activity and they have great potential for the organic synthesis. And this is because, as they have told you, that they can do many types of selective oxyfunctionalizations. They are like really promising, but for the industrial use, we need to overcome some limitations. I'm going only to mention 2 that are the important for my project, that we need to modify their selectivity, the regio, enancio, and quimio selectivity, and also we have an irreversible inactivation of the enzyme provoked by the co-substrate. To overcome these limitations, well, protein engineering techniques are used, they mainly used in the lab, and the one that many of my colleagues have talked about is directed evolution campaigns, but also nowadays they have arisen some computational techniques to do these techniques more quickly. I will focus more in FunLips because it's the one that I will talk about. And with these techniques, at the end, what we will have are new mutants or new enzymes that we will need to test to see what are the properties or what they can do. So, more specifically, FunLips is an automated method to design and run epitaxy mutations in the active sites of the enzymes by phylogenetic analysis and Rosetta atomic design calculations. In a simpler way, what we do is to select first the amino acids that we want to mutate and also to block the ones that are essential for the activity of the enzyme, well, for the activity or for the stability of the enzyme. Later on, when we have select the residues, we will run the program, and what it will do is to
compute the amino acid tolerance in each residue that we have select. That once we finish with this, we will have like what we call a constellation of enantiodivergent UPOs, in our case that it is our enzymes. So now we have our mutants. We need to determine what are the properties that have these new enzymes. So for this, what we do is a substrate screening, in this case of organic substrates, for determining the UPO footprint. That is to know what is their chemio, regio, and antioselectivity, and also their actual activity of these enzymes. In my case, I will focus more in creating regio-divergent UPOs for in the case of aromatic compounds. So more in detail, these are the two designs that we have created with fanlips, in which we have these dots are the amino acid that we wanted to mutate. These are the amino acids that the algorithm have found in all the families of UPOs, and with different results. The algorithm have give us like 15 mutants, but we have only select 7 mutants of each design. So with this, what we wanted to do is to modify the heme channel of this enzyme, because it has been seen that the heme channel, it has a great importance in the collocation of the substrate and for the activity of the enzyme. And with this, we think that we can get new activities of these mutants. To show you what we have created with these fanlips, we can see that the heme channel have been modified by different things. Here we have wider the channel. Here we have narrow the channel, and also here you can see a cavity that in the parental we don't see. So the next step is to try these enzymes and to check what they can actually do. So thank you very much, and that's it.
Well, thank you very much for the talk. So now we go next speaker, Maria Jesus Soto Alvarez from EQS Lab, and she's going to talk about lignocellulose biomass fractionation.
Hello everyone, my name is Maria, and today I'm going to talk about biomass fractionation. So, as you all know, nowadays there is an interest to find alternatives to petroleum, right? Well, the good news is that there are companies that produce cellulosic sugars from biomass to produce promising building blocks such as furfural or HMF to make renewable chemicals. So biomass has 3 main fractions: cellulose, lignin, and hemicellulose, and they have a very different composition from each other. So in order to improve the use of biomass, what we have to do is to separate these 3 fractions to process each one of them in an appropriate way. So that's why there are some challenges in biomass fractionation, like first, maximizing the yields of all fractions. For example, in a paper company, the cellulose is used, but the lignin and hemicellulose are burned. This also happens in other industry, and there are many pretreatments that are optimized to achieve high yields of only one of these fractions. So the challenge here is to achieve high yields of all 3, retaining the structure to avoid loss of value, and recovering each fraction in an easy way to apply it in different processes. Also taking advantage of minor components such as acetic acid, and of course do all this at low cost. In this way, we perform an organosolve process, which is a biomass fractionation process where we use GBL, which is an organic solvent in liquid phase and mild conditions. So our objective is to extract the 90% of the hemicellulose and lignin, and for this we put the biomass and the solvent solution in a batch reactor, and then we perform the reaction under their respective conditions. So as you can see, we obtain a 40% of cellulose with high purity, and we have worked with Stora Enso to demonstrate that this cellulose can be bleached by conventional methods and obtaining a kappa number and brightness that indicates that this cellulose has the necessary quality to use it for textile industry. And in collaboration with UJI and IATA, we have been working in a project where the main objective is to make plastics that are biodegradable for a circular economy. So the main objective of this project are that, to demonstrate that this technology, this fractionation technology can be applied to marine waste, green waste, and agri-food feedstocks. And from these feedstocks, obtain a high purity cellulose that we give it to the IATA, where they use it to make nanocellulose as a reinforcing agent for plastic. And with the cellulose of less purity, we convert it into sugars that sugars may serve as feedstock for a marine bacteria that produce PHA, which is a plastic of biological origin. And finally, demonstrate that these cellulose and sugars that haven't been used in the previous steps can be converted into levulinic acid and GDL. So we have been working with garden pruning, almond shells, and marine waste. Here I present the results for garden pruning, where you can see that we extracted the 90% of hemicellulose and lignin. That is our target. We separate the high purity cellulose from the less purity cellulose by a screening method. And as I said before, with the high purity cellulose, we give it to the IATA or to the Center of Biological Research Margarita Salas, where they use it to perform an enzymatic hydrolysis. We also obtain a high purity lignin with that has no odor and with a lower contents of sugars, ashes, and solvent. And with this lignin, we also give it to the IATA, where they use it for additive for packaging. And the cellulose with less purity, we use it to perform a chemical hydrolysis to convert it also in sugars. So finally, we have scaled up this process to a 20 liters bench reactor, where we have performed more than 30 batches and treated around 70 kilograms of wood. And we obtained the same results we got at laboratory scale, so this process is reproducible. And today we have the capacity to treat between 1 to 3 kilograms of wood and obtain 600 grams of cellulose per batch and 200 grams of lignin. So that's all. Thank you for your attention.
So last but not least, we have the last talk of this session. So Dianelis Toledo Monterrey, I think she was mentioned in some other talks, from the directed evolution group, and she would talk about ancestral sequence reconstruction of UPOs.
Yes, allow me to do one more talk about UPOs. And they are very interesting enzymes. I'm not going to, of course, to go again with the same things that my colleague has said. Just to remark that UPOs are very interesting biocatalysts because they can perform many reactions, oxyfunctionalizations, using only hydrogen peroxide as the electron acceptor. And opposite to the other monoxygenase counterparts like P450s, they don't require expensive cofactors and elaborate reaction setups. So they are very promising tools for green chemistry, biocatalysis, and pharmaceutical synthesis. So as we have seen, they are very interesting and wonderful scaffolds for protein engineering. In this case, our approach is ancestral sequence reconstruction, also a technique that has been mentioned before. That is a protein engineered computational tool that uses modern genetic data to infer and resurrect proteins for extinct organisms. Normally, Asian proteins adapt to extreme environments or evolve new functions, and the proteins that we obtain from this technique generally have enhanced stability, promiscuity, and conformational flexibility and diversity. So combining UPOS as a scaffold and ancestral sequence reconstruction, our main goal is to create even more efficient and stable enzymes for various applications. So the methodology that we follow is general for all the ancestral sequence reconstruction, and is focused in two main steps. First is the ancestral protein reconstruction, that is referred to the construction of the phylogenetic tree, and the ancestral protein resurrection, that is the production of the nodes in the modern hosts. So regarding ancestral protein reconstruction, we use 1000 extant sequences, and we perform a multi-sequence alignment in a way that we created a tree, a phylogenetic tree, and with the curation and the polishing of that tree, we obtain a high-quality phylogenetic tree that we use for the next step, that is the resurrection of the nodes. So as I told you before, the first step will be the protein expression, in our case in yeast, in Saccharomyces cerevisiae and in Pique pastoris, and the modelization of the enzymes to study how they are constructed respect to the modern counterparts, and the initial characterization in terms of stability, and also hydrogen peroxide tolerance, and also the various substrates, in this case colorimetric substrates, both of peroxidase and peroxygenase activity. So the phylogenetic tree render several nodes. We tried to resurrect 6 of the nodes, and we obtain activity in 4 of them, 2 coming from the Basidomycota and other 2 coming from the Ascomycota phylum. In terms of the initial characterization, we characterize it with different parameters like pH profile, thermostability, hydrogen peroxide tolerance, and enzyme activity. And the most interesting results in this case are the hydrogen peroxide tolerance and the enzyme activity, since normally, as Paula mentioned before, hydrogen peroxide is a hurdle for the ones that we work with UPOS, because they cause inactivation. And in the case of our nodes, all of them show higher stability tolerance with the cofactor. And also in terms of enzyme activity, some of the nodes show increased activity with peroxygenase substrates, which are the most interesting ones, and they are very promising scaffolds for next rounds of light evolution. So now we are working on the purification of ancestral variants and the parentals, so we can characterize with a broader panel of substrates, with more interesting substrates, aliphatic and aromatic ones, and a further characterization of stability, of pH and thermostability with other techniques. So I want to acknowledge my group and also our spin-off company, EvoEnzyme, and the work that does allow me to perform this work. And of course, thank you for your attention.
I think we are at the end. Probably the director wants to say some words, but I would take the opportunity that I'm here on stage to say thank you on behalf of all the more seniors in the researchers at the Institute, including myself, for the very nice presentation, for staying on time, and actually very clear, all of them, and particularly acknowledgement to those who have made the effort to present in English. I think for many of you, I know it has been challenging, and I think it's a great effort that hopefully pays off when you are invited to present your work at conferences and things like that. And as Marcos says at the beginning, I think this is a great opportunity for us to put names and faces together, see what our neighbors are doing, which is not bad. I think we all learn about UPOs. This is very good. In my floor, I know now all these faces, I know what they are doing. And thank you for the organization. Marcos, maybe you want to say something?
Sure. Thank you very much. It's time for eating. Yeah, thank you. I will use this also. So, Darío, thank you very much for joining us. These talks were a little bit shorter than yours yesterday, but I hope you can probably make some simulations about UPO, which becomes super famous today in this forum. Okay, good. So please, everybody, keep up, cheers up, and keep doing good science, and do a fantastic work now in lunch, because I saw that some of the food was not eaten in the breakfast, in the coffee break, so now you have to do a better job. And if in the end you see that there is a little bit of spare food, take a Tupperware and take it home. Okay, it's on the house. So thank you very much. Now close this session and also take a look at the posters, please.
Esmorzarem pa amb oli i sal, ho vestirem amb unes copes de vi. Deixant de banda la ciutat, la tarda és llarga i potser més.